What are coelacanths? Why would a marine fish contain a sugar that makes up the exoskeleton of insects? How do sharks sense fish? Why do we care about lamprey immune systems? Dr. Chris Amemiya from BRI studies these questions using comparative genomics. This research will improve our understanding of marine ecology as well as immunology and holds implications for both the medical and biotechnical fields. This is RadioBio.
Hey everyone, I am Jeff Lauder, a second-year student in the QSB program.
I'm Genevieve, a first year in the QSB program.
And I'm Morgan, and I'm a second-year student here in the QSB program.
And we're here with Dr. Chris Amemiya from the Benaroya Research Institute. His really broad, far-reaching research exploring everything from immunological systems of lampreys to the way sharks can sense fish, and how we can use some of these model organisms to study immunology in general. Thank you for being here, Dr. Amemiya.
Well, thank you for having me.
So a lot of your work that you described today in your seminar to the QSB department and program was focused on lamprey and the coelacanths. So why are these organisms interesting? Why do we study them? A lot of people, I think, aren't familiar with lampreys. They're actually widely distributed, even in California, but people don't even know about them. So can you tell us just a little bit of background of lampreys, why you're interested in them, and kind of how you got into researching them?
Yeah, so lampreys are very basal representatives of the vertebrates. They are, together with the hagfishes, at the base of the living vertebrates. And it's a long story of how I actually got interested in working with lampreys in the first place. This was back when I was in the Boston University School of Medicine. I was in the Center for Human Genetics there, but working on Hox genes. And in order to do the work, we had developed, when I was still a postdoc at the Lawrence Livermore Lab, we had developed methods for large insert cloning, which were necessary for the Human Genome Project. And in particular, we're developing these bacterial artificial chromosome cloning systems for cloning these pieces of DNA that are 100 to 150 or 200 kilobases in length. As it turns out, back then, there was no way to actually sequence genomes that like we have today. So we had to clone out all the pieces that we were interested in, and in particular, the Hox genes. And my collaborator at the time, Frank Ruddle, who was at Yale University, who was a pioneer in doing mouse transgenesis and for studying Hox genes, figured out that it would be a great opportunity to combine our skill sets, as it were, and to study these Hox genes. What were the Hox genes? How were they comprised in these basal vertebrates? What were their cis-regulatory elements? All these kinds of things. And in order to do that, we actually had to have these bacterial artificial chromosome libraries. Mm-hmm. So that's it was a match made by necessity, I guess. and he is Frank died a couple of years ago, but this had actually led to a couple of grant proposals, actually three that were funded to study, to use these bacterial artificial chromosomes to do the requisite sequencing. And Frank was an expert at making transgenic mice. And so the idea was to take these long stretches of DNA to insert into these pieces of DNA reporters, such as LAC-Z reporters, and then to introduce these into the mice to see where these things were going to be expressed. If they had the same sort of control that, for example, the mouse Hox genes had. And that eventually led to some really interesting findings. Of course, we expanded these to many other organisms as well. But the ability to take these pieces of DNA, to alter them, using genetic engineering techniques at that time in the yeast, now done in bacteria, and then to take those and put those into the mice and express those. That was really a key to doing the kind of evo-devo that eventually a lot of people had ended up doing. Mm-hmm. So that was my first foray into studying lampreys, and it, like I said, it occurred because we were studying Hox genes, and bacterial artificial chromosomes.
And so, just a brief injection here for people who aren't familiar. Just a quick reminder of what Hox genes are and why we study them, how they're implicated in these kinds of systems.
Right. So the Hox genes, when people study evolution, it had always been, to this very day, people are very interested in Hox genes because they contain a stretch of DNA and amino acids, about 60 amino acids that are encoded by 180 base pairs that encode this thing called the homeobox, which is a piece of, it's part of the protein that can physically bind to certain stretches of DNA, and they serve as basically transcription factors. So most of these Hox genes, and the human genome has 45 or 46 of these Hox genes. They're intimately involved in regulating many other processes. So early in development, these Hox genes are turned on. Once they're turned on, they regulate other genes in this cascade. So they're very important from a developmental standpoint. And so Frank Ruddle, who's a very prominent developmental biologist, who had discovered the Hox genes in mice, along with his colleagues in Drosophila, he had a very large interest in understanding how these Hox genes could contribute to evolutionary divergence. So this was the reason why we actually pursued the Hox genes in particular in the lampreys.
Actually, you brought up a good point. You brought back the lamprey. I'm curious, what do you use this model organism now? Before you use it for Hox genes, but what are you doing with it currently?
So, right. So that's a good question. We have many projects that have since taken off on the lamprey. Because we had these bacterial artificial chromosome reagents, we were able to collaborate also with some other folks, namely Max Cooper, who was at the time was at University of Alabama at Birmingham, and Max and his postdoc at the time, Zeb Panzer, were very interested in trying to understand the immune system of the lamprey. And I should point out that the immune system of the lamprey, it was shown several years ago, at least 40 years ago, that when you immunize lampreys, you could generate a fairly robust immune response. The immune response was similar to what you found in us or in a mouse, and one of the hallmarks was immunological memory. So several years ago, about 40 years ago, experiments were done using basically taking red blood cells, a certain type of red blood cell, for these type O blood cells, as it turns out. You could immunize with these human type O blood cells these lampreys, and the lampreys would generate this nice, robust immune response, and it was a very repeatable kind of immune response. But nobody was able to ever isolate immunoglobulins, which are the hallmarks of an adaptive immune system. So, to make a long story short, Max had a way to immunize these lampreys and to isolate certain immune populations of cells, and using a transcriptome approach. And by altering our null hypothesis, as it were, we were able to isolate what we thought were molecules that would be good candidates for being surrogates for the immunoglobulins or the antibodies. And these were molecules that had these leucine-rich repeat motifs. And that eventually led us to clone out these genes from these bacterial artificial chromosomes from the libraries that we had, and show that these, using a combination of approaches, show immunologically that these were, in fact, involved in immune recognition, and that the locus itself undergoes these massive rearrangements at the local level to generate these diversities, to generate the diversity that is necessary for combating all these antigens that are being that the organism actually sees throughout the lifetime.
Mm-hmm.
So, like you were saying before, so jawed organisms all have T cell receptors, and then the lampreys, or our outgroup, don't have these types of methods, but they still can do an adaptive immune response. So they instead use these leucine-rich proteins, like you were saying, and you were calling these ones the variable
lymphocyte receptors.
Okay,
Yeah. You're right. So there are a lot of black boxes here. We don't know. We have some ideas how the rearrangement occurs, but clearly they're doing it in a completely different way than the rearrangements that are necessary for generating the diversity for the immunoglobulins or the T cell receptors. This we know. But we think that during the development of their respective cell types, and they also have T cells that make this variable lymphocyte receptor. So B cells and T cells. So they have both lineages that we have. But the ones that are in the B cell type cells we call VLR-Bs. The ones that are in the T cell type are the VLR-As and Cs. But nonetheless, there are certain kinds of transcription factors that are associated with the development of these respective lineages, which, as it turns out, are kind of similar to what you see in B and T cell development. There are a lot of questions that we're currently trying to ask in terms of the origins of the VLRs, the development of the VLRs, etc. There's a lot of questions. In fact, the VLRs, we know based on transcriptome and some other kinds of data that they're also expressed very early in embryonic development, which seems kind of strange that these genes are being expressed and presumably utilized even before there's an immune system. So this is well before an immune system or any kind of tissue for immune recognition. They are expressing these genes, and we can find them in places that are actually, that would suggest that they're necessary for embryonic development. Now, this is all unpublished work, but I've been talking about it for a while, so it's not like a secret or anything.
Yeah. So I mean, just broadly speaking, I mean, this seems like, is there opportunities for looking into the lamprey immune response versus human immune response, and is this kind of helping us understand more of even the human immune response by studying this kind of alternative system?
Yeah, I think what the selling point for when we actually did get our NIH grant to study this is that this is a large multi-gene family that is being utilized for immunity, clearly, because we can immunize and we can get these immune responses. We can even find we can clone out the specificities against some of these antigens. But it's also being utilized in another fashion. Like I said, it probably is involved in early development, because when you knock out the VLRs early in development, as it turns out, you abrogate normal development of the lampreys. So it's a good example of a gene family that's being utilized for at least a couple of different purposes. And yes, we're pursuing this. Are there any parallels to the immune system in human? That awaits further investigation, I think.
Actually, that leads me to a different point. You're saying there might be, a different repertoire. Is there like maybe a system that we don't know of, or a network that is deciding this, or could it be an epigenetic or environmental cue? Is there something else that we're not looking into that might give us a cue of what's really going on?
Yeah, I think that's a really good question. The only answer I can say is that when we study their genes that are expressed in the earliest, when they're expressed early in development, and we sequence the repertoire is very limited, and so it seems that they're not undergoing as much rearrangement, that there's some sort of predetermined specificities that are necessary for the early developmental function. And it has been our thought that perhaps because these things are receptors, that they're serving some role in in cell lineage tracing or something to this effect, whereby it's requiring some innate signal in order to contribute to the early development. So it's some sort of developmental, like a cell adhesion molecule or something like that. But we're, like I said, we're still pursuing this line of investigation.
So there's a lot of really interesting implications there.
Yeah, I didn't even know lampreys had something resembling B or T cells, so that's pretty cool. I learned something today.
So, what is chitin, and why do we care about it? Like, what makes it so important?
Well, chitin is really simple. It's like cellulose. Cellulose is the number one polymer in the biosphere. It's made by plants, and it's basically a repeating unit of glucose. Well, chitin is similar in that it's a glucose derivative. It's called N-acetylglucosamine, and it's found very prominently in the natural world. It's the second most prevalent polymer in the natural world, and it is the most prevalent polymer in the aquatic environment. And so, in terms of just the sheer amount of, from an ecological standpoint, it's the fact that you're finding chitin turnover at about anywhere from 10 to 20 gigatons is spectacular. Wow, because if you don't, if you didn't turn over this chitin, if you didn't have, and the only way you're going to turn it over is by using, effectively using these chitinolytic enzymes, which are found in bacteria. If you didn't have these things, the ocean would be one very thick slurry. Okay, so it's an important molecule for a lot of reasons, and of course, it's found on horny exoskeletons of all these arthropods, as well as in fungi. It was first found in right. So, in fact, one of the diagnostic things that they look for in the clinical lab for when you have a fungal infection of the lung is you look for chitin. It's a very simple assay. So we use those same kind of simple assays to study chitin in the organisms for which we found chitin synthase genes, which were absolutely surprising because, and where I'm talking about the fishes and the amphibians, these were not thought to have chitin. And if you go online, I mean, almost every source will tell you that there's no chitin in any of the vertebrates. And in fact, in the literature, it's considered dogma that chitin is not in any of the vertebrates. So this was kind of surprising finding. And
so sorry to interrupt you, but just to touch on that. So what is the fish that you first kind of discovered this chitin in, and how did you kind of again get to that point of studying this fish?
Yeah, so we didn't necessarily use a chitin per se. What we did was we were able to show when we looked for genes that were actually missing in the mammals, but they were found in the fishes. Mm-hmm. We found a handful of, or actually about 50 of these genes. And as it turns out, one of these genes, which was an unknown gene in NCBI, as it turns out, this gene was most closely related to chitin synthase. Mm-hmm. Chitin synthase is the final step in chitin biosynthesis. It's a kind of a complicated process, but it is the final step. And so, and because it was so highly related to the invertebrate chitin synthesis from Drosophila or from shrimp or what have you, it was really quite surprising. And we had to therefore figure out whether or not these chitin synthases were making chitin in these vertebrates. And as it turns out, they are. Mm-hmm. And so it's our mission now to figure out what they're doing. It's certainly not making horny exoskeletons, because based on the way the chitin is actually distributed, they're not in these really hard structures. Mm-hmm. Most of it seems to be in some sort of a gelatinous kind of structure.
Mm-hmm.
We find it in the fishes and the amphibians.
Mm-hmm.
We do not find them in the so-called amniotes, which are the reptiles and including birds,
Mm-hmm.
and the mammals.
Mm-hmm.
But we do find them in some tetrapods. We find them in things like amphibians and axolotls and frogs. And so chitin per se, based on the physical characteristics of chitin, we find, well, it's known that chitin is less soluble in aquatic solvents. And you can take chitin, and for example, you can buy chitin from Sigma, these shrimp shells, and you can try to solubilize them. They don't really go into water very well. And you have to basically treat them in a certain way with acids or bases to get them into water. And then they'll come out of water really easily if you're not careful. So it's really a mystery to us why it is that the chitin is even in these organisms and how they're actually existing. And maybe that hydrogel that is forming, clearly they're in some sort of mucoid form, is protective. We know that chitin can serve as an antibiotic. We know that chitin, as you say, is less soluble, but it also serves as filters. For example, there's this structure called the peritrophic membrane in Drosophila and many other invertebrates. It actually serves as a filter to protect the organism from toxic waste in their gut, for example. We don't know. I mean, this is like really, really new stuff, but based on what we've seen and the distribution of the chitin, especially in the integument, which is the skin of fishes where we've done most of the work, we can see that it's forming this very mucus-like structure, which may be involved in the extracellular matrix, which is absolutely critical for cell-to-cell communication.
And so specifically what organisms are you studying in this case and finding the chitin in? So during your talk, you mentioned the coelacanths.
Yeah, so again, we've been lucky because we found in every case where we've looked and found chitin synthase genes in their genome, we find the presence of chitin. And we've lots of these examples. Lots of fishes have chitin, and they express them in the same way. We find them in the gut, we find them in the skin, etc., in the eye. But most of the work, experimental work, has been done on the zebrafish, because zebrafish, you can get embryos and you can do a lot of experimental biology, and a lot of there are lots of lines available that have for which you have fluorescent markers in these lines. So it makes it a lot easier to actually study. If you're studying, for example, we think chitin is involved. We see it early in development in the endoderm. So there are some endoderm markers, fluorescent lines for the endoderm that we can get our hold of. And we can do the requisite experiment. For example, we can knock down the chitin synthase genes and see what happens to these cells, for example. Those are the kinds of experiments that are very basic, but they can tell us a lot about the potential function of what chitin might actually be doing.
You just kind of talked about specifically knocking out chitin genes. Can you go over specifically what happens when you knock out those genes? So what are we seeing in these zebrafish or coelacanths or the organisms that you're using when you actually do those gene knockouts?
Yeah, so we can't do any experiments with the coelacanth.
Yes, yeah.
Yeah, because coelacanths are endangered, and they're also quite large. And so those experiments, I mean, we were really, really lucky to be able to sequence the coelacanth genome because, just getting material, it's on the appendix of the CITES endangered species. Mm-hmm. In any case, when we knock out the chitin synthase genes using either this CRISPR technology or using morpholinos or chitin synthase inhibitors, such as these pesticides that have been developed to prevent insects from forming a one of these horny exoskeletons, we see some the embryos really don't develop normally. You have all kinds of effects on the eye, on the axis, on the skin, and it doesn't form a gut normally.
What kind of effects would chitin have on the eye? Like, why do you think it's actually affecting the eye development anyway? Like, what's going on there?
Yeah, that's a really good question, and we really haven't pursued it as thoroughly as we should. This is one of those areas that we don't have really a lot of expertise on the eye, so we have to collaborate with someone on this. But the places where chitin is found, or the place where chitin is found in the eye, is surprising. So we see it in all the eyes of of every single fish that we've looked at, as well as amphibians, such as axolotl. And the place where chitin is found is, in particular, on the rods and the cones, there's a structure called an outer segment. Mm-hmm. Now, the outer segment in these photoreceptor cells these structures are some ciliated structure, at least evolutionarily, and they have all these like cristae in them, and in between all these little layers are the opsins, and there are many different opsins, right? Each photoreceptor is expressing a different opsin gene. So opsins are these proteins that are involved in sensing light in a particular wavelength, because they're when they are exposed to light in a particular wavelength, then they can resonate, and that eventually fires that particular photoreceptor to generate a signal, right? So if there's a photon in the right wavelength, whether it be for the rods or the cones, you get a signal, and ultimately, of course, that signal goes to the optic nerve, and it goes.
Perception of a color, or
the perception of a color is what actually allows also the spatial resolution, the topographical map, right? And so what it's actually doing in the outer segments is anyone's guess. And of course, why it should be the case that knocking down the chitin in these will give rise to basically a defect in eye formation is anyone's guess. But
I'd like to move to the opposite end of the animal. You were talking a little bit about in developing fishes, or you'll have a fecal pellet that'll have a lot of chitin in it. Do these pellets kind of continue? Does it create an envelope during development? And do they ever stop after the first fecal drop? Or do they continue throughout development and they continue throughout the fish's life?
Yeah, so that's a good question. And I probably didn't specify what we actually see. I don't think the chitin is actually sloughing off. I think it's part of the tube at the anus. However, it's clear to me that it's enveloping this fecal pellet, and we know that, of course, when they drop the fecal pellets, that they don't have chitin except for the chitin that's in the fecal matter itself. Okay, the sheath is actually, it stays on the fish, I think. But clearly, that purpose of actually, so as you know, being ecologists and whatnot, fecal pellets are really, really important for the ocean. They form the marine snow, not zebrafish because they're freshwater, but then we have evidence that most, or of the ones that we've looked at, the marine organisms, the marine fishes, they have chitin that surrounds the fecal pellet. Now, why that's important is that people have done these studies of fecal pellets, and if the fecal pellet doesn't sink, then it won't go down to the bottom of the ocean. Of course, as you know, that the fecal pellets are really important for the biomass in these really deep zones. So it has to sink, and I suspect that the chitin is involved in compaction of these fecal pellets.
I'm little weighted bricks, essentially.
Yeah, no, exactly. So when you look at when you look at these schools of fishes and you see the fecal pellets, like herring and things, they all drop to the bottom, right? So they have to be more compact than the surrounding environment.
I personally want to thank you for your time here in the lab of RadioBio.
Yeah, this was a great discussion, so thanks for taking part in this.
Well, it's always fun to talk about science, It's my favorite subject.
Thanks, guys. This is RadioBio.
Signing off.
Have a good day.
RadioBio is supported by the Quantitative and Systems Biology Graduate Group at the University of California, Merced.